What is dengue? So this is the genome size and accession number of the dengue virus. So now let's check out the structure of dengue virus. The dengue virus consists of lipid bilayer, capsid, envelope, and RNA genome. These are the closest related organisms to Zika virus, West Nile virus, and yellow fever virus.
Now we take a look at the original research paper of the dengue virus. This is the phylogenetic tree of dengue virus that were prompted by Mega software.
Hi, I will be presenting tools in genome analysis and its advantages and disadvantages. First of all, we have RT-PCR, the reverse transcription polymerase chain reaction. RT-PCR is used to convert viral RNA into complementary DNA, and they amplify specific genomic regions. Its main role in the method is to detect and amplify genome fragments before sequencing. The advantage of RT-PCR is its high sensitivity, making it ideal for detecting small amounts of RNA. However, it requires precisely designed primers, and even small amounts of contamination can lead to false positive results. Next, we have Sanger sequencing, the ABI3130 DNA sequencer. After amplification, the DNA is sequenced using the Sanger method, specifically with the ABI3130 sequencer. This step is crucial for producing high-quality, accurate sequence data that will be used for genome assembly. The key advantage of Sanger sequencing is its exceptional accuracy. It's still considered the gold standard for short DNA sequencing. On the downside, it is labor-intensive and not ideal for sequencing entire genomes in bulk due to its low throughput. Then, the third tool we have is MASER, the multiple sequence alignment tool. MASER is a software tool used to align multiple DNA or protein sequences. In this study, it helped align dengue virus genome fragments and compare them across different isolates. Its strength lies in being fast and accurate, especially for large datasets. However, it may be less effective when aligning highly divergent sequences or those with complex RNA structures. Lastly, we have MEGA 5.03 version, the Molecular Evolutionary Genetics Analysis. MEGA software was used to construct phylogenetic trees from the aligned sequences. It played an important role in analyzing evolutionary relationships between dengue virus strands. The advantage of MEGA is its user-friendly interface and the flexibility to choose from multiple phylogenetic models. But its limitation is that it's not ideal for large-scale datasets or advanced Bayesian modeling, which require more specialized tools. In conclusion, these four tools, RT-PCR, Sanger sequencing, MASER, and MEGA, form a comprehensive workflow for genome analysis. Each one plays a unique role, and understanding both their strengths and weaknesses helps ensure accurate and meaningful genetic analysis of viral pathogens.
Next, let's go to process of getting gene bank result and comparison among genes. In this study, researchers analyzed dengue virus DNV1 strains isolated from viremic patients in Delhi between 2009 to 2011. Importantly, they also included a historical reference, the prototype Indian DNV1 strain from 1956 P23086, to compare how the virus has evolved over decades. The first process of getting gene bank result is RNA extraction and RT-PCR. The process begins with extracting viral RNA from patient serum samples. To decode the full genetic blueprint of the virus, researchers use overlapping RT-PCR primers. This method ensures complete genome amplification by generating multiple overlapping fragments like piecing together a puzzle. The second process is sequencing and genome assembly. Next, the amplified DNA fragments were sequenced using Sanger sequencing, a gold standard method. However, due to technical limitation of the primers, the very end of genome 5 and 3 terminal regions couldn't be fully captured. Despite this gap, the team assembled high-quality sequences for analysis. And the last process is gene bank submission. All assembled genome sequences were submitted to GenBank, a public NIH database that stores genetic data for global research access. While the access numbers aren't shown here, they allow other scientists to download and study these sequences further. From RNA extraction to GenBank submission, this workflow highlights how viral genomes are decoded and shared, key step for tracking outbreak and designing treatments. Now, we have covered how many the viral genomes were sequenced and submitted to GenBank. Let's dive into the genomic analysis. This study compared key genes in DNV1 strains, focusing on two major categories: structural proteins, essential for virus architecture, and non-structural proteins, critical for viral replication. We will also explore how mutation at the nucleotide and amino acid level impact the virus. First, let me go to the differences of structural and non-structural genes. First, the structured proteins, which is capsid, pre-membrane, and envelope. Mutation in this protein were analyzed for their impact on antigenicity, the virus ability to recognize by the immune system. The E protein is especially important. It's the virus key to entering host cell and the primary target of antibodies. Changes here can influence how well our immune system fight the virus. Beyond structural proteins, the virus relies on non-structural proteins, which is NS1 to NS5 to replicate. NS3 acts as a multi-tool. It's both a protease cut viral proteins and a helicase unwind RNA for replication. NS5 is the virus RNA polymerase, copying its genetic material. Positions under positive selection were identified, suggesting these regions are evolving rapidly, likely due to immune pressure or adaptation to hosts. And next, let's dive into the differences of nucleotide and amino acids. Nucleotide level changes. At the genetic code level, strains from North versus South India show up to 5.2% divergence in their nucleotide sequences. This regional variation hints at how the virus adapts to different environment or host population. But not all genetic changes alter proteins in amino acid level changes. Synonymous mutation, silent changes in DNA that don't affect the protein sequence. Non-synonymous mutation alter the protein's amino acid, potentially changing its function. For example, a mutation in NS5 might make RNA replication faster, helping the virus spread more efficiently. In summary, structural protein mutation, especially in E, can help the virus evade immunity. Non-structural proteins, which is NS3 or NS5, drive replication and are hotspot for evolution. Nucleotide diversity across India underscores the need for region-specific surveillance. Understanding this difference helps us track outbreaks, design vaccines, and develop targeted therapies.